Be with you in just a minute. Thank you. All right. So anyway, vehicle transmission again is an inanimate reservoir. So airborne, waterborne, foodborne. Again, I mentioned waterborne is a big one, especially in third world countries where the sewer is their drinking water. So a lot of diseases are passed that way. And probably the biggest of these is foodborne. You know, food poisoning, foodborne illness, that's a big problem, even in this country, because people do not take precautions with their food like they should. You see a person here cutting chicken on a cutting board. If they come behind that and cut something else and it doesn't get cooked, then that can transmit salmonella and other bacteria that way. Chicken is one of those, like pork, that you really need to be careful with. They carry a lot of diseases. Beef isn't so bad, but chicken and pork are the ones that typically, out of the meat groups, are the ones that carry the most diseases. A lot of it has to do with how they're raised. You got to think of chickens and chicken houses. They're packed in. They walk around in their own poop all the time. Pigs are the same way. They lay around in a pigsty and their own poop all day long. So it's easy for them to pick up different diseases and things and carry those, and then that can be transferred to you in the raw meat. But if you thoroughly cook everything, you should be fine. Um, but you do need to make sure you get that internal temperature where it needs to be on that. Right. Now, vectors are arthropods. These are things that can pick up a disease from one person and carry it to another. Fleas, ticks, mosquitoes. In fact, a lot of your disease projects are probably going to cover diseases that are transmitted by vectors. Things like Lyme disease and malaria, and all of these are carried by insects. And these can be transmitted in one of two ways. Mechanical transmission, you take a fly and he goes in the backyard and lands on a pile of dog poop, and then he comes and lands on your food at your picnic. He can pick up bacteria and then bring it to you and transmit that to your food. The other is biological transmission, where something takes the blood from one person or animal and then takes that and bites another person and transmits that way. And this can be transmitted either by bites or by feces. And we think about some of our household pests like roaches. Roaches are notorious for leaving feces behind that have a lot of bacteria. Roaches and roach poop can cause asthma in children. They can cause all kinds of disorders. So that's one reason we try to keep them out of our homes, other than they're just, disgusting to look at. But they do transmit disease as well. So we need to make sure that they are under control. We're all going to have roaches and flies and stuff at some point in our house, but we just don't want it to be an infestation. If we get one or two here or there, that's one thing. But when you have hundreds living in the walls and living everywhere, that can be a disease vector. Right? Video is going to go over the mosquito and show you how it's a vector and the different species that can carry different diseases. Now, these are mainly tropical diseases that we deal with mosquitoes, but there are some that are transmitted in, the region where we live. We see in West Nile and Zika, and those things start popping up around here. So again, the mosquito is and for a little trivia question for you, the animal that kills more people every year than any other is the mosquito. And yes, it's an insect, but it's also an animal. So that one is the one that kills the most, and it's by disease, and it's by transmitting these diseases. So let me pause myself, and then we'll watch this little short video on mosquitoes.
Come on, You should go to T-Mobile and they'll give you a brand new iPhone 16 on them.
Go to T-Mobile and get iPhone 16 with Apple Intelligence on us on our most popular plans. Plus, get up to $800 when you switch.
Ugh! Get out of here, you stupid mosquito! Ugh! I don't want your stupid diseases! Hey guys, Julia here for D-News. Mosquitoes are often seen as one of the most annoying creatures on this planet and arguably one of the most deadly. These buzzing nuisances carry deadly diseases like yellow fever, dengue fever, lymphatic filariasis, and famously, malaria. According to the World Health Organization, malaria infects over 500 million people a year and kills 3 million. So why are mosquitoes such good carriers of disease, especially malaria? Well, interestingly, hashtag not all mosquitoes. Of the 3,000 species of mosquitoes, only 3 of them seem to be the ones spreading most of the human diseases we're familiar with. Anopheles mosquitoes carry malaria, filariasis, and encephalitis, and inflammation of the brain. Culex mosquitoes carry encephalitis, filariasis, and the West Nile virus. And Aedes mosquitoes carry encephalitis, yellow fever, and dengue fever. In fact, many species of mosquitoes aren't good carriers of disease at all. According to Dr. Kenneth D. Vernick, a microbiologist at the University of Minnesota, most mosquitoes are malaria resistant and the susceptible ones are the oddballs. Most mosquitoes actually fight malaria with their immune responses to parasites. After a mosquito sucks up its bloody meal, a parotrophic matrix or barrier forms in between the bug's lining of their midgut and the blood. While it can take a few hours to form, it does trap some parasites inside, possibly providing protection against some of them. The malaria parasite doesn't infect mosquitoes at all. It never enters their circulatory system. Another response mosquitoes have is to coat the parasite in a melanin capsule, but this comes at a cost. The mosquito has to commit resources to coat the invader, which takes resources away from things like reproduction, resulting in the infected mosquitoes becoming less fertile. So though the parasite can enter the mosquito's body, it often doesn't make them sick, which is good news for the parasite or virus because if their host dies, they die too. And malaria's next trick is getting from the mosquito into a human body, probably yours. The parasite uses some of the very things that help a mosquito feed to easily enter the next host. Mosquitoes have substances in their saliva that weaken blood vessels, promote bleeding, and shut down the human body's natural first lines of defense. These evolutionary advantages help invaders slip into their next host. With an injection directly into the bloodstream, they bypass a lot of immune defenses, and some parasites actually change a mosquito's behavior to make it easier for them to achieve this goal. One study in PLOS One found that malaria makes humans smell more attractive to mosquitoes, and another study published in the American Journal of Tropical Medicine and Hygiene found that malaria makes mosquitoes feed longer. It invades the mosquito's salivary glands while in the sporozoite stage, which probably helps the parasite get into the next host easier. But in their invasion, they interfere with the mosquito's saliva production, which the mosquito needs to locate blood vessels in a host. With impaired salivary abilities, it takes longer for the mosquitoes to feed, to get the same amount of blood, which is just fine with the parasite. It gives them more time to jump to their new host. So though mosquitoes are good at ferrying parasites like malaria to our bloodstream, they don't benefit from their morbid task, and instead put themselves in more danger and can become infertile. The saying goes, don't kill the messenger, but in this case, you should totally kill the messenger. I agree with one Cornell University researcher who called mosquitoes the most vile and useless species on the planet. Why don't we just get rid of them all? Well, Trace and Natalia find the answer in this episode right here.
A world without mosquitoes sounds like it doesn't suck. But what purpose do mosquitoes actually serve? Don't they have to have a purpose? Not really, actually. They exist because they haven't gone extinct yet.
Alright guys, so how do you feel about mosquitoes? Would you kill them all or do you think they're useful in some way? Let me know down in the comments below. Don't forget to hit those like and subscribe buttons and keep coming back to D-News so you don't miss a single episode.
All right. So that gives you an idea of how mosquitoes transmit disease. Again, they are a vector. A vector is just something that carries things from one place to the next. So again, that's just a way of transmitting the disease. And again, the animal itself, the mosquito, really doesn't get affected that much by it, other than a few little things like the saliva production we heard about. But they don't get the disease. They just simply carry it. And they transmit it from one place to the next. And we see this a lot with different vectors, with ticks and other parasites. that suck blood. There's some parasites that have kind of hijacked that and use it as part of their life cycle. One of the most interesting classes I ever took was parasitology and looking at how parasites evolve and their life cycles. It's amazing they're even here. As many hosts and things as they move through. Sometimes they'll go through a larval stage in one organism, then they have to be transmitted to another one and then to another one to become an adult and never reproduce. And it's just amazing that that whole transmission actually happens. But they've developed ways to do that over millions of years. Now, another thing that we worry about with infections is hospital-acquired infections. And these are nosocomial infections. And 1 in 25 hospital patients will acquire some type of nosocomial infection. That's a pretty high rate of transmission, but you got to think you're in a place where you're around tons of sick people. If you work in a hospital, over time, you will become immune to almost everything. At first, when you first start working, you're probably going to get sick a lot. You're probably going to contract viruses and things like that. But once you've acquired those, and you get better, you're going to have antibodies against all of this stuff. I saw the same thing when I first started teaching. My first year teaching, I got sick quite a bit, and I hardly ever get sick. I'm one of those that if I get sick, it's usually something bad. It's rare that I actually get, I've never had the flu, for instance, but it's rare that I contract anything. My immune system's pretty good. But that first year, I had colds all the time. You're just exposed to so much. In a hospital, especially, you're just dealing with tons and tons of sick people. So it stands to reason that things are going to be transmitted. Now, about 2 million people per year are infected, and about 70,000 deaths occur because of nosocomial infection. So these are very serious, and it's something to worry about, and it's something you need to take precautions against. And when you go to work at a hospital, they're going to give you the protocols that they make you go by to keep things sterile, to keep you from transmitting things from patient to patient. Again, sometimes people cut corners and they don't do what they're supposed to do and they spread things. But if you follow the guidelines and you wash thoroughly and that kind of thing, hopefully you won't be transmitting these along the way. Right? So these are going to result, these hospital-associated infections or healthcare-associated infection, just because there's microorganisms in the hospital environment. Again, you're around a bunch of sick people. Also, patients typically are weakened already, so their immune system's already kind of shot. So when they come in, they're more susceptible to other diseases that they may come in contact with. And then just the chain of transmission. Just think of a nurse. She goes from room to room to room, and if she doesn't take precautions, she's going to take whatever's in this room to the next room and the next room and the next room. And you may be one of those that carries some of these diseases, but you may be immune and you may never get sick, but you may transmit it to someone else. Right? So again, in hospitals, we have a lot of compromised hosts where people already have problems, and these diseases are opportunistic and they just find a way to get in. So things like burns, for instance, you've opened the skin up. Now you're just wide open to the outside world and anything can come in. If you remember from anatomy, we talked about the first thing with burns is you worry about fluid loss, but that second thing is going to be disease because they simply have no first line of defense and their body's wide open. So it's easy for these organisms to get in. So healthcare associated infections, again, the compromised host, the chain of transmission, the microorganisms in the environment all lead to this. And again, you have to take a lot of precautions to make sure that these are kept to a minimum. They're going to happen, but keeping them to a minimum is the thing. So if we look at the most, the most acquired types of diseases that we see in a hospital, UTIs about 13%, surgical site infection 22%, respiratory infection, lower respiratory 22%, gastrointestinal infestations or infections, Clostridium or C. diff causes about 12% of these. That's a big one that, again, hospitals, if you've ever smelled that, it's the most god-awful smell in the world. Someone has a C. diff infection, but it's very common. So again, about 12% of the 17% are just C. diff patients, and then bloodstream infections, and then others. So there's all kinds of different things can be spread through disease. Bacteria tends to be the most prevalent one. If you notice, almost all of these are bacterial infections, right? So when we look at these, the viruses aren't the ones you worry about quite as much as the bacteria, because a virus doesn't have the ability to stay outside of the human body for very long, at least in most cases. But bacteria do. And so bacteria can easily be transported from one person to the next by another person, like a nurse, right? Or, even anybody that goes and visits room to room, even people that clean up after the patients and things, that they can spread infections as well, because they're dealing with feces and things of that nature. So it's easy for that bacteria to be spread around, a little bit harder for the viruses to be spread through the hospital. Okay. So these are the most common bacteria that we see. Again, you can look down through here. Most of these are probably familiar to you, at least now they are, since you've had lab and you've worked with a lot of these, like Pseudomonas you worked with in lab, Staph aureus you worked with in lab, Staphylococcus, Pterococcus, the enterics, those you've worked with too. So Candida, that is the one that causes yeast infection. So that's the one fungus we have on there. There's E. coli. We've heard of that. You've used that in lab. We use Klebsiella in the Murphy lab. I don't know. I mean, I'll do in my class, but I don't know if they've used that one with you or not. But again, these are just some common microorganisms associated. Now, the precautions you need to take, basically, there's standard precautions. These are the minimum practices, the basic stuff. And then there's transmission-based precautions that you supplement to those standards, all right? So contact precautions, droplet precautions, airborne precaution, again, wearing masks and things of that nature to help prevent transmission. Pretty much common sense. I mean, if you know anything about disease transmission, it's common sense to do these things. Wash your hands, disinfect anything that you use with patients, like a tub that you use to give a sponge bath. Instruments need to be autoclaved and cleaned scrupulously. Disposable materials, get rid of it, incinerate it. They're infected. So again, most of these are just common sense things that you would just normally do as a human being to begin with. You do this around your house if you had someone that's sick. So again, not rocket science there. All right, now we've talked a little already about emerging infectious diseases, and these are diseases that are new, increasing in incidence, or showing a potential to increase in the near future. Most of these are zoonotic, which means they come from animals. Most of these are viruses, and most of these are vector-borne. We are starting to see some new infections coming out now, and we're seeing some old infections come back. And we'll look at some statistics here in a few minutes and some graphs to show you a few of these. But if you look at this map down below, you can see all of these different diseases that are popping up around the world. And again, we are interconnected now, where we fly back and forth. So what happens in one place eventually will happen somewhere else. And again, viruses, especially the RNA-based viruses, have the unique ability of being able to mutate fairly rapidly. All right. They can mutate from a form that infects an animal to a form that infects a human. I just read today, as a matter of fact, when I was at the dentist waiting to go back, they've now detected bird flu in many different mammals and even humans. And so now it's moving from birds to mammals. And if it makes that jump and it starts to sustain itself in a mammal population, then we are next. And they are really worried about that moving through that stage of going through into a mammal and then into a human. We're mammals after all. So if it can attack other mammals, it can mutate very easily and attack us. And it's already infecting people. And it's just not infecting them at a high level. But again, all it takes is that one mutation, and it allows it to enter the cell easily, and then it's game on. And the flu is easily passed from person to person through droplets in the air, just like COVID was. So If it does start infecting humans, then we'll have to look into that. So when we look at emerging infectious diseases, there's some contributing factors. Genetic recombination. Again, these bacteria and viruses have the ability to mutate, and they can change into different forms. Remember, E. coli lives in our gut, and it's one of our normal microbiota that lives there. But one form mutated to form a toxin, and that is the O157 strain. And if you get it, it causes bloody diarrhea and it makes you very ill. There's new strains like Vibrio cholerae, which causes cholera. This is another one that is mutated into a form that's different from the old one. And again, we're not going to have much immunity to those new forms of the bacteria, or in the case of flu, a virus, because we've never seen it before. That was the problem with COVID. We had never encountered it, and our immune system just wasn't primed and ready for it because we had never seen it, right? Hantavirus, this is one that's been in the news too. If you remember Gene Hackman and his wife that died mysteriously. Of course, he was very old. He was like 95, but his wife looked 64. But she died of hantavirus, which is pretty rare. But somehow she contracted that, and usually it's contracted from mice, deer mice. And so somehow she come in contact with rat droppings or mice droppings and caught that virus, and it ended up doing her in. At least that's what they come up with after the autopsy. Right. So again, modern transportation is the big one. We move a lot. We move all over the place. So it's easy for these viruses and things to move around. The insects, again, we looked at the mosquito earlier. It's easy for them to carry things from one person to the next, or from an animal to a human. Ecological disasters, war, just us expanding our settlement into places like the rainforest exposes us to new diseases. And then, if we don't control some of these vectors like ticks and things, then they can spread things like Lyme disease. So there's ways, I think I showed you this the other day, but they're, basically sterilizing mosquitoes down in the Keys of Florida, and they are making them where they only have males. Males do not bite. It's only the female mosquito that needs your blood, and needs your blood to fertilize its eggs, or basically make its eggs viable so that they can hatch. So the male doesn't have anything to do with biting you. But they've shifted that population from, basically 50-50 to almost all male. And now those mosquitoes aren't biting anyone. Pollen's killing me. I had a fishing tournament Sunday, and I've been out on Sunday all day. They pretty rough on me yesterday and today. So we look at these diseases. A lot of these are going to occur naturally, of course. They're just mutations, things that occur in nature. Public health failure sometimes can cause these. We just, deal with pollution, and especially in third world countries, again, when they dump sewage into the rivers and the people drink out of it, it's easy for these diseases to spread. A newer one we have to worry about, I say newer one, we've actually used this for years in warfare, but bioterrorism, using diseases against others. And this has been done before. In fact, we mentioned when the Spanish invaded South America, they didn't really have to fire a bullet. They killed everybody with smallpox. They figured out they weren't immune to it, and they intentionally spread it, and it wiped out almost all of the native peoples. And they took their gold, and took over the land. And again, that was the first real form of bioweaponry, or using some kind of biological agent to attack someone. So that's something that we really have to worry about. Now, this is going to go over some of the most toxic things in nature, and a lot of these are produced by microorganisms, all right? And he's going to go over this with you. It's good timing, too, because my voice is about to crack. So let me grab some water real fast while this is on, and then I'll come back.
It's a story straight out of a Bond film. On September 7th, 1978, Bulgarian journalist Georgy Markov, a communist defector working for the BBC, was walking across the Waterloo Bridge in London when he felt a slight sting in his right thigh. He looked behind him and saw a man picking an umbrella up off the ground. By that evening, he had developed a fever. 4 days later, he was dead. The cause of death? Poisoning. Markov had been assassinated using an umbrella rigged to fire a pellet laced with one of the most powerful natural toxins in the world, ricin. Ricin is one of the deadliest natural substances that is not found in an animal. It's found in the beans of the castor plant. Just 500 micrograms of the stuff, that's 5/10,000ths of what a paperclip weighs, is enough to kill a man. And as potent as it is, ricin isn't even close to being the most poisonous natural substance on Earth. There are poisons that lurk in bacteria, plants, and fungi all over the place, and they're not just found in exotic tropical locales or like 1970s London. They're pretty much everywhere, and the fact is, they're there for good reasons. But what is it about their chemical makeup that makes them so poisonous? How do their toxins attack the human body with such deadly efficiency? And why does my thigh suddenly feel all tingly? Is that an umbrella? Not only is it morbid to list the most deadly toxins in nature, it's also nearly impossible. There are a lot of plants and fungi and bacteria out there capable of killing you if eaten, inhaled, or injected. And there are also a lot of variables that make it hard to quantify exactly how poisonous something is. Everything from the age of the victim to the antibodies in your system can affect how potent a substance is. There is, however, one exception. No one disputes that the most lethal toxic substance known to man, natural or otherwise, is produced by the tiny bacterium Clostridium botulinum. You probably know it by the disease it causes, botulism. Just how deadly is the botulinum toxin? One way experts use to measure the lethality of a poison is a method called the lethal dose 50%, which is the dose required to kill half the members of a tested population. Since, for obvious reasons, you can't run these tests on humans, toxicologists usually get this number, often called the LD50, by using an endlessly unfortunate creature, the laboratory mouse. LD50 is measured in a ratio of milligrams of toxin to kilograms of body weight. And in the case of the botulinum toxin, it's about 1 nanogram, a billionth of a gram per kilogram. That means that a crystal of botulinum the size of a grain of sand would be enough to kill 9,600 people. The bacteria are actually really common, and the spores they produce are found in soil and water all over the world. The problem starts when they germinate into active cells, at which point they begin to produce 7 different toxins, 4 of which are deadly to humans. C. botulinum thrives in low oxygen environments, and the 10 to 30 cases reported each year in the US are usually the result of eating improperly canned food, since canning removes oxygen from food to preserve it. But if the food isn't heated properly, the spores can stick around and set up shop in that can of Texas chili. Botulinum spores can also be found in honey, and if you eat them, they can find a nice oxygen-free nook of your body to hang out in. Kids and adults can ward off these spores before they germinate, but infants cannot, which is why babies under 12 months old should never eat honey. The spores are poisonous because they secrete a neurotoxin as a waste product. It works by blocking the release of a neurotransmitter called acetylcholine, which controls muscle contraction. This effectively makes every muscle in the victim's body go limp. Within several days, without antitoxin, the victim will asphyxiate from the inability to breathe. But yes, thanks to science, and all of those poor lab mice, there is an antitoxin. But why in the name of Mathieu Orfila would a bacterium evolve to fart the deadliest poison on Earth into its host? The answer remains a mystery. One theory is that the toxins are not aimed at the host, but rather at competitor bacteria. They create conditions where benign bacteria cannot survive, giving the evil bacteria a chance to spread. The host, in this case a human, is often just in the way of this process. So compared to the botulinum toxin, cyanide is like eating Play-Doh. Well, not really. Do not eat cyanide. Not only will it kill your face, it's also one of the most rapidly acting poisons in the world. It's found in cigarette smoke, burning plastics, also a surprising number of natural foods, including almonds and the seeds of apricots and apples. The LD50 of sodium cyanide, a common form, is 6.4 milligrams per kilogram of body weight, or over 6 million times less deadly than botulinum. But that said, it's been a popular and effective tool of death for everyone from Emperor Nero to Agatha Christie. Cyanide is a carbon atom, triple bonded to a nitrogen atom. What makes it such a fast killer is that it binds to proteins in your cell's mitochondria, the tiny power plants that keep your cells going, and keeps them from using oxygen, basically asphyxiating the body at a cellular level. It is true that some fruits and nuts contain cyanide. Apple seeds, for instance, contain a chemical that releases cyanide when it comes into contact with digestive enzymes. This is a good way to deter critters from digesting up all your seeds if you're a fruit, but there's not enough poison to kill them or us. Though I guess I wouldn't make a point of, eating a bowl of apple seeds. The good news is that if you happen to suffer from cyanide poisoning, there's always vitamin B12. The B12 that you get at the store actually contains cyanide as part of its gloriously complex molecular structure, so high doses of a precursor to the vitamin can literally pull the cyanide off of your poisoned mitochondria, and then you just pee out the poison in the form of B12. Now, back to our Bulgarian friend. Well, the castor plant that killed him is next on our list. It's native to East Africa, but grows pretty much anywhere it's warm, including the southwestern United States. The toxin it produces, ricin, is a protein found in the castor bean, but the poison itself is made from the mash that's left over after grinding the beans into oil. When the oil is heated, the ricin protein denatures or changes shape, which renders it harmless. So the toxin doesn't end up in the oil itself, which is used widely as a laxative, or used to be. Not so much anymore. Because gross. But when inhaled, ingested, or injected, untreated ricin is readily absorbed by the body's cells. Once inside, it inactivates the ribosomes, which manufacture proteins that your cells need to survive and reproduce. And once the protein train stops, life stops. Just a single ricin molecule is capable of inactivating 1,500 ribosomes per minute as it kills the cell. You may feel the effects within a few hours, but ricin is generally a slow-acting poison, taking 1 to 3 days to cause death. Hence, its popularity with assassins over the years who want to distance themselves from their crime. The tiny platinum plug found in Markov's leg had 2 tiny channels drilled into it, each able to hold about 1/5 of a milligram of the poison. But that was more than enough to kill Markov or any other human. And the toxicity of ricin can change depending on how it enters the body. Taken orally, its LD50 is about 20 milligrams per kilogram of body weight, about 8 castor beans worth of the poison. But if it's injected, like the way Markov got it, it's just 1 tiny microgram per kilogram. Antidotes to ricin, sad to say, are still in the early stages of development, but because ricin takes several days to kill, compounds are being investigated that can block it from reaching ribosomes in new cells in the body. And strangely enough, ricin is also being investigated as a possible cancer treatment because of its ability to trigger that protein-stopping self-destruct mechanism in cells, including, of course, cancer cells. Now, if you ever come across a strychnine tree, and yes, there is such a thing, my advice is to stay as far away as possible. It's native to India and Southeast Asia, and its bark contains a poison called brucine, but the real lethality comes from the tree's round, green to orange fruit, which contains the alkaloid strychnine. Tests on both sad pigeons and sad rats have pegged the LD50 for strychnine at between 3 and 14 milligrams per kilogram when taken orally, which puts it in a similar category of lethalness as ricin. But the threat here is a neurotoxin that affects the nerves in the spinal cord that control muscle contraction. Essentially, it blocks the chemicals that control nerve signals to muscle. By blocking that chemical, strychnine puts your muscles in a state of constant stimulation, causing seizures until the point of exhaustion and respiratory failure in as little as 30 minutes. This does not sound like a pleasant way to die. Finally, no roster of natural poisons would be complete without a visit to the kingdom fungi, because although few of its thousands of species are poisonous, some of the ones that are contain toxins that would give botulism a run for its money, called amatoxins. The 2 most dangerous of these mushrooms contain the most lethal poison of its kind, called amanitin, and they have the not-so-cryptic names of Deathcap and Destroying Angel. The oral LD50 of amanitin-containing mushrooms is between 0.1 and 0.2 milligrams per kilogram, so we're talking at least 30 times deadlier than strychnine. Amatoxins are essentially rings of amino acids that interfere with enzymes that, again, are responsible for the production of life-giving proteins in your cells. But here, they specifically target your liver and kidney cells. Part of the reason they're so dangerous is that symptoms are delayed for 6 to 24 hours after ingestion. So victims only know there's something wrong when their liver and kidneys begin to fail, causing vomiting and diarrhea, which is often mistaken in emergency rooms as the flu. another reason the poisoning is so often fatal. Most of these deaths in the US are from the misidentification of mushrooms by immigrants from Asia, where these particular species of fungi don't exist and instead have quite edible lookalikes. In cases that are caught early enough, massive doses of milk thistle extract can stop the worst of the damage. The extract contains chemicals that block the uptake of the toxin in liver cells, but in the worst cases, the victim can only be saved by a liver transplant. Now, I should probably stop, now that I'm just completely paranoid about everything from strange men with umbrellas to all of the formerly beautiful mushrooms in the world. The good news is that scientists and lab mice have been working hard at developing antitoxins to these most poisonous natural substances, so that if we do come across one of them, we might not die. And in a very few cases, these terribly toxic substances may even be used to save us one day by targeting cancer cells. So thank you for watching this episode of SciShow. If you have any questions, comments, or suggestions, we're on Facebook and Twitter, and of course, down in the comments below. And if you want to keep getting smarter with us here at SciShow, you can go to youtube.com/SciShow and subscribe.
All right, so this gives you a few toxins. I know some of those were plant-based, but there were several bacterial-based toxins. So those are the ones we have to worry about. Now, the study of when and where diseases occur is called epidemiology, right? And epidemiology basically is the study of disease and how they spread. So epidemiologists are going to determine the etiology of disease or where it comes from, identify other important factors concerning the spread of the disease, and then find ways to control the disease, and then assemble data and graphs to outline the incidence of disease. Now, your epidemiology project covers this. So that's what you're going to be doing with the little story that I gave you and the data I gave you. You're going to make a graph and you're going to show things. Just follow the directions and you should be able to figure out exactly what you need to do with that. All right. But we'll show you some graphs here in a second and kind of similar to what yours are going to look like. All right. So we've studied these guys and ladies before, but basically they were the first people to start studying diseases. So John Snow, remember we watched a video about cholera in London in the drinking water. And then Semmelweis, we talked about hand washing way back in the first chapter. And then Florence Nightingale, basically, everyone should know that one bit. But she looked into typhus and how it spread and sanitation practices to prevent that. All right, so if we look at epidemiological graphs, we can pull some information from it. So if we look at Lyme disease, for instance, this is from 99 to 2016. Some of this data is going to be kind of old. Some of it will be new because I just updated some of it to 2025. So we can look at actual trends with some of the data. This is some old data again that they put on here, but you can see that in the years from about 07 to about 2010, we started seeing a big uptick in Lyme disease. And what they have to do is look at these graphs and say, okay, why did this happen? Well, when they went back and looked at a lot of data and studied a lot of things, they started noticing some weather patterns. And for these years, the weather was considerably warmer in the northeastern United States than it normally is in the wintertime. And they think from that maybe the ticks weren't killed off as readily in the colder weather, and therefore more survived, and then more were out there to be able to carry Lyme disease. And that's why we saw this uptick here. So we can look at these numbers and we can say, okay, that's fine. Well, it went up to 35,000 cases in 09, but it's the why behind it. Why did that happen? And you have to look at other data sets and things to figure that out. And that's why this is such a hard thing to do. It's because you have to kind of think outside the box and think about why would this disease increase in these years? And the first thing you would think of, well, okay, what spreads the disease? That's ticks. Where do they mainly live? In the Northeast. Well, what would keep their numbers high or make their numbers higher than normal? Well, they started looking at weather patterns and they found a correlation between them. So that's what the epidemiologists kind of have to do. They have to figure out why these numbers shifted, why they are showing the trends that they're showing. Same thing with Lyme disease during a year. If we look at it, We say, okay, January, February, everything's kind of low, and then as it starts getting warmer, we start seeing more and more cases. Well, there's more than one reason for that. Number one, how many people are out in the woods camping in upstate New York in January? Probably not very many, right? You may have a few hunters out in the woods and that type of thing. The other side of it, ticks aren't as active in these months, right? So when people start entering the woods and ticks start becoming more active at the same time, it makes sense that we would see a peak in the summertime. It just makes sense altogether because more people, more ticks, more Lyme disease. So it's a pretty easy correlation to pull off of that one. We look at this one, this is tuberculosis. And if we notice right here, right after World War II, we had a lot of tuberculosis cases. Number one, we didn't understand exactly how it was spread, but through the war effort, we learned a lot about it, right? And we learned that in people that live in close proximity, tuberculosis spread very rapidly. So army barracks, for instance, if one person brought it in, almost everybody would get it. Prisons, same thing. You put people in prison and they're boxed in together, they're going to spread this disease very easily. It's an airborne bacteria, all right? But once we learned about it and we started taking precautions, we see a rapid decrease down. Now, I'm going to show you another graph of TB or tuberculosis here. And right now, we're starting to see an uptick in tuberculosis, right? So back in 1992, it's been going down ever since, right? But all of a sudden, in the last couple of years, we started to see an uptick. The problem is tuberculosis has mutated and it's become resistant to antibiotics. And that's the reason they think it may be going up here is because the treatment, you can't treat it as well. And people are able to contract it and spread it. And also during the mutation phase, the bacteria became virulent, which means that it attacks people more readily, and they think that may be a part of it. So we're starting to see a little bit of an uptick. I know this only goes through 2022, but this has continued. And I read an article the other day about TB making a comeback. So that's something we're seeing there. All right. Now, measles, you've probably heard about this in the news recently. We started with the vaccine back here in the 60s. And if you ask your parents about it, they all got their measles shot. And well, my parents, not your parents, most of you. But back then they got their shot and it was a miracle for them because they would all get measles as children and it could be deadly, very deadly. So the vaccine was introduced and look at what happened. Boom, measles cases went down. We had a little bit of uptick here and there. And then the second dose of the vaccine was recommended. And by 2000, we had essentially eliminated it in the United States. It was gone, right? Didn't see measles anymore. Now, though, look at what's happening. People have stopped getting vaccinated to measles, and now we're seeing upticks in the disease. Now, part of this is due to people from other countries that are unvaccinated coming in with the disease. But now we have a lot of Americans that are against vaccination and their kids aren't vaccinated and they're starting to contract it from those people. So we're seeing kind of a double-edged sword there. And we've seen a big uptick. If you've studied anything in the news lately, you know, Texas has had a big problem with measles cases and they're still rising. And we're starting to see that. So again, we can look at these graphs. If we look back here, you know, COVID vaccine was developed back here around 2021 or close to it is when it was administered. And that's when people stopped vaccinating. They started having these anti-vax type, you know, feelings or whatever, and they stopped vaccinating. And we're starting to see these upticks in cases because people aren't protected anymore. Again, we've essentially eliminated it from the United States. It was gone. It didn't happen anymore. It only happened in other countries, but again, now with influx of people from other countries and people not vaccinated, we're starting to see cases go up. Okay, so with epidemiology, we have a couple of branches. We have descriptive epidemiology, and that's collection and analysis of data. Analytical epidemiology, that analyzes a particular disease to determine its probable cause. And then experimental epidemiology involves a hypothesis and controlled experiments. This is where people go through clinical trials to try to see what we can do about a disease or how we can control this. So the CDC analyzes epidemiological information, and we give morbidity and mortality rate. I remember during COVID, people kept getting these two confused. All right. Morbidity is the incidence of a specific notable disease, all right, or notifiable. And we'll get into what notifiable means in a minute. Mortality is the deaths from notifiable diseases. So morbidity is how much disease is out there. And then mortality is how many deaths actually occur. And people kept getting these two confused. And I remember people arguing left and right about COVID and stuff. And it's like, you don't even really know what you're putting out there because you don't understand what these terms actually mean. Now, notifiable infectious diseases, this is a disease in which a physician has to notify the CDC and tell them that this person has had this disease, right? So again, back to morbidity and mortality. Morbidity is a number of people affected in relation to the total population in a given time period, whereas mortality is the number of deaths from a disease in relation to the population in a given time. So two different things. Morbidity, basically how many people have the disease. Mortality, how many people die from the disease. And again, if you mix up those numbers or intentionally mix those up, you can make the mortality rate look a lot worse by using the morbidity data and saying that's mortality. And I saw that over and over again, even in the news with the press. They just didn't understand the difference between these two numbers. And they would go on, the nightly news and tell the morbidity rate and act like it was the mortality rate and how many people were dying. And they got it mixed up. Right? Now, this is a list of all the diseases that doctors have to notify the CDC about. So if anybody has these diseases and comes into their office, they have to report it, right? It's a pretty long list. And some of these, again, are just diseases that you may not think much about. You're like, well, if I got that, I can take an antibiotic and get rid of it. So let's say you go in with gonorrhea and you take some antibiotic. Well, you're getting reported to the CDC. They're going to tell them you had gonorrhea. Now, they're not going to use your name or social security number. They're just going to report a patient had gonorrhea so they can get the good numbers of how many people have this disease, right? And again, that's what they're doing with this information. They're trying to see, okay, are these diseases increasing, decreasing, staying the same? And then look at, again, the morbidity rate of these to see, okay, is this disease increasing in number? Or do we have more people getting this disease? So you may hear something in the press about gonorrhea increasing in a population, or syphilis, or some of these other STDs. That's where they get this information from, is these notifiable diseases and the reporting that's done on them. All right. Now, if we look at a particular disease, MRSA, this is one that you really have to worry about spreading in a hospital, right? So this one is methicillin-resistant Staphylococcus aureus, or MRSA. There's two strains that we really worry about. This is the one in healthcare systems, is the USA 100, and then the strain USA 300 is the one that's community acquired. That's where you get it outside of the hospital. Now, I don't know if you've done this yet in lab. You might have, I'm not sure, but if you haven't, pay attention to this because you'll need to know this for your lab. But when we look at MSA, and this is an agar that has specific qualities about it, all right? So selected media, when we talk about it, and I think I mentioned this before, but we'll go over it again, selects for one particular organism. So in this case, this has a high salt concentration. So Staphylococcus can tolerate salt. Most other bacteria can't. So any Staphylococcus is going to grow on here, all right? So it's going to select for Staphylococcus, but it's also going to be differential in that it differentiates between S-MAR and S. aureus. All right. So S. aureus produces an acid when it ferments, and that causes the media to turn yellow. The S-MAR does not produce the acid, and that causes it to stay pink. So when you look at this, you can select for the Staphylococcus, and then you can differentiate between the two different epithets or species of Staphylococcus, right? And you'll do a lab like this where you'll use MSA, and then you'll use EMB, which is another media, and you're going to select for different organisms. And in this case, you're going to select and differentiate. All right. Now, this is something I want you to do for extra points. I'm going to give you 3 extra points just for turning this in. So let me go over it with you. And then I've got a little quiz for you to do afterward. But let me go over this. That can come up. So, it's not working. It worked early for me. Oh, there you go. All right, so this is put out by the CDC, and it's almost like a little game, right? But what I want you to do is go through, and you can do level 2 is locked until you get double level 1. You don't have to go through and do all 12 in level 1. So you can just pick any one you want from level 1. Here, click on that. Right? And then you're going to pick any one of these you want, and you're going to solve the outbreak. When you get done, just take a screenshot, and I'll put a Dropbox up for it. And again, this will be extra credit for you. All right. So all you're going to do is hit continue. And it's going to give you a set of clues, right? And it's going to ask you about certain things or tell you certain things, and you're going to have to pick down here. Oh, crap. Already selected it, but well, let me go through and I'll show you what to do with this, right? So this one says, if I can start this over again. Cut that off so we can hear. So you're going to select one of these. So go to level 1. I'll go through and we'll read the clues. All right. So it's going to tell you startling outbreak has been reported in Chicago and Milwaukee. Within the past 2 days, 15 people have visited emergency rooms complaining of high fever, fatigue, coughing, chest pain, difficulty breathing. Several people have died and others are near death. Your help is needed to end this outbreak. You'll begin this investigation as a trainee. To become a novice, you'll need to review each clue, answer the questions correctly, and solve the outbreak. All right. It said, I accept this mission. All right. And here it says the first reported case is a 37-year-old musician from Chicago named Zeke. You start by talking to Zeke and find out what he was doing the night he got sick. Zeke said he started coughing and feeling very tired. Shortly after, he led a music workshop for 50 students at a Chicago concert hall. He thought he might have picked up something from one of the students in the class, or perhaps he caught it 2 days ago in a flight home from a vacation in Africa. So you got a couple of clues in here. One, he went to a music workshop with 50 people. He also took a plane trip from Africa on the way home. So it's going to ask you, where do you think Zeke caught whatever made him sick? Was it the flight home from Mali? Was it the music workshop? Or is it too soon to know for sure? What would you think you would pick here? Just put it in the chat box if you have an idea of which one. Too soon? I agree with that. Okay, so too soon? Yes, too soon. All right, so it says right. Yep, good. All right, so it says you earned 100 points for this question. So we'll go to the next clue. So you were correct about that because there's no way of knowing what happened, right? He could have got it anywhere, not even one of those places. I'm sure he went other places too. So next clue says more of the mysterious illnesses are being reported in Chicago and Milwaukee hospitals. Patients, loved ones are in a panic and hoping we can help find out what made everyone sick. Lab results show patients are infected with a bacterium that doctors suspect might be anthrax. You immediately send the samples to public health laboratories for testing, but that will take a couple of days. In the meantime, you need to talk with everyone who is sick and their families to find out which activities they participated in before becoming sick. If you look at the responses to your questions, it seems that many of the sick people participated in the same activities recently, like working in a government building or attending a music event. So what would you do next? Recheck the data to see if there's one activity that all the sick people have in common, interview workers in the government, or cancel the music events in Chicago. And he says recheck. I agree with that. Click that. Looks like it's good. Again, you'll go to the next clue. It's going to give you more information, and then you'll just keep going on. Now, don't do this first one since we've already kind of went through it. But you see, it's not that difficult, right? So just go back and pick one of the original ones, and get my score. Right. So just go back and click level 1. Select any of the other 11 that are left, right? So don't do this first one. All right, but you can pick from this one, this one, this one. Just kind of hold down your mouse in between them and it'll let you go through. So find one that you might be interested in. It seems like it would, be kind of a cool thing to go through and try to figure out. But you can go through these and it gives you some initial information stuff here, where the location's at and all of that kind of stuff. All right. I will put this link up for you on Blackboard, and then I'll put a Dropbox for you to put your screenshot in. So when you get done and you get your certificate, just take a screenshot of it. Again, I'll give you 3 extra points on your final exam or your final test. All right. So just go through that and perform those. Let me pull back up the PowerPoint. All right. We're going to watch this next time. You'll see that it's pretty long, but this is going to go over all the different bacteria and viruses, the stuff you're doing your disease project on. You haven't come up with a project idea yet. Thursday, you might be able to watch this and get some really good ideas because it's going to go through a bunch of different microbes, all right, and tell you what diseases they cause and that type of thing. All right. So let me pull this down. And your quiz should be set up for you. So go ahead and go to Blackboard, and don't remember, we haven't done one of these in a while. But I only had one person actually send in a thing that I've seen, because it usually sends it to my email. I've only seen one. Somebody done that? All right. Yeah, I saw yours today. All right, I'll go take a look at it. Usually it emails me when you send one of those dot forms in. I haven't seen any except for the one today. I saw that one. I'll look into it and I'll let you know Thursday which one. But those are all really good projects. So hopefully y'all picked a good one out. But anyway, go to your daily quizzes. It's daily quiz 6. It should be open until 7. There you put it out. Yeah. I'll look back and see. I can go to the jot form and look at it. That'll be for your next test, the 3 points. Final test that you're going to take. You're welcome. Does anybody need this open later than 7 tonight? Some of you have work and stuff going on. Everybody able to get this done by 7? Ready? You need a later date? Change it to 8. Will that be good to everybody? Due date, time. I hate that word also. These conditions first. Oh. Things set up for 8, so you should be good to go on that. Covers chapter 14. All right. If anybody has any questions, I'll stick around for a minute and answer anything. If not, we're good to go. Take your quiz, and we'll see you Thursday. Getting close to the end now, guys. Yeah, the epidemiology project, I moved it up a little bit. That's the only one. I think the rest of them should be fine. I did have to adjust the syllabus a little bit. So just go by the calendar on Blackboard, and you'll be right on it. Yes, that is the part of the group project. You'll have the oral part, which will be your video, and then you'll have a pamphlet to go with it, half and half. Yeah, the epidemiology is separate. That's just for this chapter. Yes, the due dates on Blackboard are correct. So don't look at the paper syllabus. Go by the calendar on Blackboard. You're welcome. I looked at that today. I thinking it might be a little confusing. Got anything? If not, I'll close this on down.